Welcome back everyone. This is our final module of the omics internship that we're focusing on machine learning for genomics. So in this video, we are starting with supervised machine learning in R. This is an important step where we will move to building predictive models. Our goal is to train algorithms that can learn from labeled data. For example, predicting whether a sample belongs to one class, cancer, or another normal based on its feature, or in our case, we are working with gene expressions. You already know the basic idea behind the supervised learning. So in this session, we will focus on how to set up the workflow in R, from data preparation to model training and evaluation. So let's get started and see how machine learning helps us turn data into real predictive power. Before we jump into coding, let's take a quick look at the main steps that make up supervised learning workflow. No matter which algorithm you choose, whether it's a random forest, HBM, or artificial neural network, the process usually follow the same core stages. Step 1 is the data pre-processing. Before training any module, we need to make sure our dataset is clean, consistent, and well formatted. So in this first step, this includes checking for missing values, normalizing the data, and make sure all variables are in the right structure for further analysis. Next, we split the data into training and test sets. The training set helps the model learn pattern, while the test set checks how well the model performs on unseen data. So a common split is around 70 to 80% for training and 20 to 30% for testing, although this can vary depending on the dataset size. This step is important in machine learning because it helps us estimate how our module will behave in real world scenario, not just on the data it was trained on. After that, the third step is the model training. This is where our algorithms like random forest, SPM, or neural networks actually learn from the training data we provide. But before training our model, we will prefer to perform feature selection, which helps to identify the most important feature or variable that drive the classification task. After training our model, we evaluate how well our models perform. We can assess model performance based on different matrices like accuracy, precision, recall, and ROC AUC curve to understand the model strength and weaknesses. So in R, we use a carrot package to handle all these steps because a carrot package provides a unified and reproducible workflow for pre-processing, training, and evaluation of machine learning workflows. There are other frameworks and packages are available like MLR, but we will stick with carrot package for simplicity and consistency throughout this module. So for now, our main focus on the classification models, where the goal is to predict categorical outcomes such as whether a sample belongs to a disease or control group based on its gene expression. Now let's move into R and begin preparing our data. We will have to pre-process the data before we start training. This might include exploratory data analysis to see how variable and samples relate to each other. For example, we want to check the correlation between predictor variables and keep only one variable from that group. For this purpose, we often use unsupervised methods such as PCA and principal component analysis or clustering for this purpose. Some algorithms might be sensitive to data scales or outliers. We should deal with those issues in this step. In some cases, the data might have missing values. We can choose to remove the samples that have missing values or try to impute them, because many machine learning algorithms will not be able to deal with missing values. So before training our module, we need to address our missing values. So first of all, we need these few packages that will help us with the data handling, pre-processing, model training, feature selection, etc. Now let's load our data set. We have a matrix. We have a data set with 486 genes as a feature and 462 samples. This is a batch corrected and processed expression data. It's mean we already pre-process and normalize our microarray data. Let's look at this data. Here you can see we have samples as columns and genes as raw names. So this is our gene expression data that we will use for this machine learning module. You may use the dataset from your previous module you work with. You can either work with your differentially expressed genes to evaluate their performance based on machine learning models, or you can use the entire clean dataset you created earlier. So at this point, our dataset is ready for initial preprocessing. We have columns as samples and genes as rows. The first thing we will do is check for normalization and transformation. We want to ensure all samples have similar expression value and distribution. Let's look at how expression values are distributed across samples. Box plots are a quick way to check the normalization. We already perform this step in our quality control. Seems like there was some normalization done on this data set because gene expression values per sample seem to be have on the same scale. However, looks like they have long-tailed distribution, so a log transformation may fix this problem. First, let's visualize how log transformation affects the data distribution. I have taken one sample from our data set, specifically this sample number 8. Sample number 18, just to show you how the transformation changes the shape of the data. We will plot two histograms side by side. So see the difference. Before log transformation, the data is right skewed. After transformation, it is more symmetric and comparable across the genes. We can now apply the transformation to the whole data set by using simply this log10 function. Transform our data set. Here this +1 is the pseudo count that handles log 0. That is +1 inside ensures that we don't take log of 0. Next, machine learning models expect feature as columns and samples as rows. So if our data has gene as rows, we need to transpose it first. In base R, we have function T for transpose. We use it to transpose our data. Now, each row represent one sample and each column represent one gene. So now we have the data in a proper format for machine learning. After that, we can filter predictor variables which have low variance that are not likely to have any predictive importance, and they will just slow our algorithms. Previously, we filtered out our data based on prob IDs. Now again, we can check at gene level, like do we have any low variance expression in our data or not. The caret preprocess function can help filter the predictor variables with near zero variance. Another option is to select the topmost variable genes manually, like top 50 or top 1000 based on the standard deviation, or we can filter the predictor variables that are highly correlated. As the fewer predictor variables we have, the faster the machine learning algorithm can be done. You may choose not to do this step, as some machine learning algorithm can handle correlation between predictor variables. Next, for better numerical stability, we need to center and scale the data. When we center our data, we subtract the mean of each variable from its values. So after centering each variable, for example, each gene will have a mean of 0. This step helps make all variables comparable in terms of their position on the numeric scale. And when we scale the data, we divide each variable by its standard deviation. That means after scaling, each feature will have a standard deviation of 1. And this center and scaling step ensures that the feature measure on different scales. For example, one gene with expression value of around 1000 and another around 2 contribute equally to the model performance. So together centering and scaling improve the numeric stability of machine learning algorithm, and are especially important for distance-based or gradient-based models, such as support vector machines and neural networks that needs data on scaling. So, without these steps, feature with larger numeric ranges might dominate the learning process and skew the results. Another problem in the real world data set, missing values are the common thing. We have lots of missing values in our genomes data. In genomics data, this could be happen due to failed probes or other technical issues. So you already learned how to deal with missing values for micro array data analysis, and for machine learning it is also important to handle missing values. Here in our data set we have 47 missing values. So here we have two options. We can remove them, like you may want to discard the samples with missing values or discard the predictor variables like feature and genes. But removing genes or the samples both have downsides. You might be removing the predictor variable features that could be important for the prediction, or removing samples with missing values will decrease the number of sample in the training set. So another option is to impute missing value. Imputation can be as simple as assigning missing values to the mean or median value of the variable. Or for the more precise imputation, the another method is KNN imputation. In this case, the algorithm finds samples that are most similar to the sample vector with NA values. Then the algorithm averages the known missing values from those neighbors and replace the missing value with that particular value. Here we will use this KNN impute method with the carried preprocess function. So again, let's check if we have any missing value in our data or not. So at this time, we don't have any missing value in our data set. We need to make sure our target variable or the column that represent the outcome or class is properly set up and merged with our gene expression data. In our data set, we have two classes, cancer and normal tissues. So we want to use this column as our target variable, which tells the algorithm what to predict. So first we will create a vector, group vector containing these class labels. Then we will merge it into our expression data set so that each sample has its corresponding class label. So here we have our groups in a factor and levels as 1 and 0. And to make this compatible with most of machine learning algorithm, we will convert these cancer and normal class labels into a 1 for cancer and 0 for normal. And we set the factor levels as 1 and 0 because many machine learning algorithms in R treat the first level as a positive class. In our case, that means cancer will be considered the positive class for machine learning. Finally, we will bind these group labels to our expression data to create a single data frame containing both predictor variables, gene expression value, and response variable, this class labels. So here, if we look at our data set, now with our gene expression value, we have another column for a class label or groups. Now, this is the data set that we will use for machine learning. Further, data partition is the most important part of machine learning workflow. We split our data into training and testing sets using stratified sampling to maintain the sample portion of classes in both subset. We will use 70-30 split, meaning that 70% of the samples will be used for training, 30% will kept aside for testing. This approach is considered the gold standard for evaluating model performance because it allows us to test the model on data it has never seen before, give a more realistic estimates of how machine learning model will perform on new unseen samples. Here, how we can do it in R. Setting a random seed by using set.seed ensures that the results are reproducible. That means you will get the same split every time you run the code. So here we will use caret create data partition function from a caret package. Here we specify the group, our group from this data. We want to split on the basis of our target group, and p equals to 0.7 ensure the splitting and 70-30 splitting. So after that we pass this train index to our data. This is our main data to create a training data set. So here in our training data set we have 324 samples for model training, and to create test data we need to subtract this train index from our actual data. So in this way, in test set we have 138 sample to check the model performance that we will use during model evaluation purpose. So here we will perform one more step for machine learning to avoid any overfitting and data leakage. We extract gene expression value from our training set and target column. We separate out our target column from our gene expression value. So our X train data contains only gene expression values for training set, and Y train contain only labels for our labels for training set. Similarly, X test contain only feature of training of test set, and Y test contain the labels for test set that we will use in further machine learning algorithm. In this way, our dataset is now cleanly divided into training and test set that is ready for further machine learning training and evaluation. So that's it for today. In this session, we discussed the supervised machine learning workflow and completed our data preparation. So in the next session, we will move on to the feature selection, where we will apply Boruta and recursive feature elimination to identify the most predictive genes. So make sure your data is ready for the next session.